This is Cellular Respiration by Matthew Herons and Gabe Biondo. In this video, we will be discussing the process of cellular respiration. Cellular respiration is a means by which cells extract energy stored in food and transfer that energy into ATP. The equation for cellular aerobic respiration is C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy. This energy is usually ATP. So ATP, and aerobic cellular respiration usually produces between 36 and 38 ATP. There are two types of cellular respiration: anaerobic and aerobic cellular respiration. In aerobic respiration, glycolysis is followed by alcoholic fermentation or lactic acid fermentation. If oxygen is present, this will result in aerobic respiration. In aerobic respiration, glycolysis is followed by the citric acid cycle, commonly referred to as the Krebs cycle, the electron transport chain, and oxidative phosphorylation. So if oxygen is present, pyruvate will go to the citric acid cycle, or acetyl coenzyme A will, and the citric acid cycle is followed by oxidative phosphorylation and the ETC. In glycolysis, one molecule of glucose is broken down into two 3-carbon molecules of pyruvate or pyruvic acid. This releases 4 molecules of ATP. We say that glycolysis only produces 2 ATP because there is both an energy investment phase and an energy payoff phase. 2 ATP molecules are used in glycolysis in the investment phase, and this results in the production of 4 ATP molecules. The 4 produced molecules minus the 2 used molecules equals a net gain of 2 ATP molecules being produced. Glycolysis can be represented by the equation two ATP plus 1 glucose yields 2 pyruvate plus 4 ATP. Glycolysis occurs without oxygen and produces pyruvate, which is the raw material for the Krebs cycle and fermentation. During glycolysis, ATP is produced by substrate level phosphorylation, which is the direct enzymatic transfer of a phosphate to ADP, forming ATP. So a phosphate is added to ADP, and when the phosphate is added, the diphosphate goes to a triphosphate, forming ATP. After glycolysis, the next step in aerobic cellular respiration is the Krebs cycle. But before the Krebs cycle occurs, there needs to be a prep stage. This is the prep stage. In the prep stage, pyruvate is converted into acetyl coenzyme A, and pyruvate is also known as pyruvic acid. Acetyl coenzyme A combines with OAA to produce citric acid, and the citric acid is then able to enter the Krebs cycle. Acetyl coenzyme A and pyruvate are not able to enter the Krebs cycle. So pyruvate combines with coenzyme A to form acetyl coenzyme A, which produces 2 molecules of ATP. So there are 2 pyruvates made from 1 glucose. So that means there's 2 NADH produced and 2 acetyl coenzyme A's produced. After the prep stage, the Krebs cycle is now able to occur. And the Krebs cycle is the same thing as the citric acid cycle. The citric acid cycle completes the oxidation of glucose to carbon dioxide. Carbon dioxide is given off during cellular respiration, and it more specifically is given off in the prep stage of the Krebs cycle and the Krebs cycle itself. The cycle turns once for each pyruvate, so twice per one molecule of glucose. The cycle generates 1 ATP per turn. By substrate level phosphorylation, this means it generates 2 ATP in total. The remainder of the chemical energy is transferred to NAD plus and FAD to make NADH and FADH, which are reduced coenzymes. They carry higher energy electrons to the electron transport chain. Overall, the Krebs cycle releases 6 NADH, 2 ATP, and 2 FADH2, and CO2 as a waste product. So we can see here that it is a cycle, and the NADH, the FADH2 all leave and enter the electron transport chain. The next step of aerobic respiration is the ETC, or electron transport chain. The electron transport chain is a proton pump and couples two reactions. It uses energy released from the exergonic flow of electrons to pump protons against the gradient from the matrix to the outer compartment of the mitochondria. This establishes a proton gradient. The ETC makes no ATP, but it sets the stage for ATP productions in chemiosmosis. So here is the ETC. It consists of proteins that allow the H plus ions to exit into the outer compartment of the mitochondria. So the protons exit, forming the proton gradient, and NADH and FADH2 drop their electrons off into the ETC. The ETC carries electrons delivered by NADH and FADH2 from glycolysis in the Krebs cycle to oxygen, which is the final electron acceptor, through a series of redox reactions. In a redox reaction, one atom loses electrons or hydrogen, which means it is oxidized, and one atom gains electrons or hydrogen, which means it is reduced. Oxygen acts to pull electrons through the ETC. Most of the energy produced in aerobic cellular respiration is made in the process of oxidative phosphorylation. This is the phosphorylation of ADP to ATP by the oxidation of the carrier molecules NADH and FADH2. Chemiosmosis uses potential energy stored in the form of a proton H plus gradient to phosphorylate ADP and produce ATP. In oxidative phosphorylation, protons are pumped from the matrix to the outer compartment against the concentration gradient. So here we can see the concentration gradient, and they are pumped through the ATP synthase, which spins, and this phosphorylates ADP into ATP. The protons are only able to diffuse down the concentration gradient and into the matrix via the ATP synthase. As protons flow through the ATP synthase, they generate energy to phosphorylate ADP into ATP. Oxygen is the final hydrogen acceptor, combining half an oxygen molecule with 2 electrons and 2 protons, forming water. So we can see water here, which is a product of cellular respiration, and we can see ATP, which is also a product in cellular respiration, usually makes 32 to 34 ATP per molecule of glucose. So that's all aerobic respiration. So now let's move on to anaerobic respiration. Glycolysis is still the first step of anaerobic respiration. The next step is fermentation, which takes place in 2 forms: alcoholic fermentation or lactic acid fermentation. So here are the 2 types of fermentation. One is alcoholic on the left, and on the right we have lactic acid fermentation. For fermentation to occur, there needs to be an adequate supply of NAD plus to accept electrons during fermentation. Here are 2 types. Let's talk about alcoholic fermentation. Alcoholic fermentation is the process by which cells convert pyruvate from glycolysis into ethyl alcohol or ethanol and carbon dioxide, and usually occurs in yeast cells. So in this process, NADH is oxidized into NAD plus and sent back to glycolysis. So this occurs in yeast cells. NADH releases its electrons, which helps convert pyruvate into ethanol, and then NAD plus goes back to glycolysis. The next type of fermentation is lactic acid fermentation. In lactic acid fermentation, pyruvate from glycolysis is reduced to form lactic acid or lactate. So here's our pyruvate made from glycolysis, and we have two of those, and that is reduced into 2 lactate, one for each pyruvate. And in this process, NADH gets oxidized back into NAD plus and gets sent back to glycolysis. So NADH releases its electrons, forming NAD plus, and that is sent back to glycolysis. So that is both aerobic and anaerobic respiration, and I hope you enjoyed.